Good morning, everyone. So, welcome to the third online lecture series. In the lecture series, the third talk, I would say, today we have among us yet another eminent speaker, a young full professor, as you see. So, it's Professor Prashant S. Kharkar. Welcome, sir, to this
Thank you.
online series, and thank you for accepting our invitation. Dr. Ashwini Chauhan from the Department of Microbiology had been instrumental in organizing this talk. And today, on his behalf, I'm here to welcome you on behalf of the entire department. Our audience are mostly our students, our scholar, our faculty member, and also those from outside our institute because it's on YouTube Live. So, now for the participants, a brief introduction about our speaker who actually needs no introduction whatsoever. So, still, it's customary, and so I would be introducing Professor in a short while. So, about our speaker. Is it clear? I hope you can see it. So, Professor Kharkar, he completed his master's and Ph.D. in pharmaceutical chemistry from Institute of Chemical Technology, that's ICT Mumbai, in 2000 and 2004 respectively. He did extensive postdoctoral research in medicinal chemistry in USA, and in 2019, Professor Kharkar joined the Department of Pharmaceutical Sciences and Technology at the Institute of Chemical Technology, that's ICT Mumbai. Professor Kharkar's research area are wide and they are all very much relevant, and they include design, synthesis, biological evaluation, and preclinical trial of new chemical entities as therapeutic agents, study of biofilm forming and coron sensing inhibitors for gram-negative bacterial pathogens, discovery of potential antitubercular agents targeting the MTBGUAB2, the discovery of novel cancer cell line cancer stem cell inhibitors, then computational prediction of the toxicity of small molecules, and development of novel fluorescent probes for biomedical applications. So, this is just in brief to tell you the specialization of our guest today. And without any further delay, I would not stand between him and you all. So over to Professor Kharkar for his presentation. Please, sir.
Uh, Thank you so much, ma'am, for the kind introduction. Good morning to one and all present here. Today, I'll be talking about a very interesting and very advanced technique called cryoelectron microscopy. Let me just share my presentation. Is it visible?
Yeah, it is.
Okay. Okay, perfect. I'll just put it on this full screen mode. Please let me know if the slides aren't changing, because sometimes that happens with Google Meet. But then I am sure this is a new other platform, so probably it should be okay. So, students and faculty who are present here, like I mentioned, I'll be talking about cryoelectron microscopy, a cutting-edge technology for 3D structural determination of biomolecules, particularly proteins. Although the technique is evolving, the hardware as well as the electron microscopy part of it, so it has undergone radical changes since last decade or so. The technique is not old, not new, I would say. It's pretty old. It dates back to early 1970s when somebody tried to use electron microscopy for looking at the structural 3D structural aspects of biomolecules. In fact, one of those guys, Dr. Frank, he eventually got Nobel Prize along with two other guys in 2017 for chemistry. And I will be mentioning that towards the end of my talk as well. right? So I'm sure some of you, or probably all of you, might have heard about cryoelectron microscopy and the way it is taking over the structural biology part of the life sciences. It's really amazing. In fact, in 2017, I went for a conference, Gordon Research Conference on computer-aided molecular design in the United States. And then this conference is a very focused conference, like all Gordon conferences are. So on the last day, the last lecture was by Dr. Sriram from one of the UC, that is University of California campuses, and he spoke for 45 minutes on cryoelectron microscopy, and his lab is actively involved in that. And that 45 minutes were so stunning that he stole the thunder of all 5 days of conference just by 45 minutes. And that's where I actually got very much interested into cryoelectron microscopy. Not that I'm a structural biologist or something, but I'm at the application side of it, like where we use the three-dimensional structures of biomolecules for drug discovery or drug design aspects or drug design applications. So that's exactly why I thought this is, since this is a lecture series on life sciences, and students should know the details of this particular very upcoming technology, and it has a great potential. As you can see, somebody like three scientists got Nobel Prize for this, and many more probably will get in future, right? So before I proceed with my talk, it's just 18 or 18 19 slides, so and then 45 minutes I should be done. There are some interesting videos, so hopefully you will enjoy watching those video. And I tried to make it very very simple, so that it doesn't become kind of a technical jargon for you, right? So before I proceed with my talk, I would like to thank organizers, Dr. Ashwini and Dr. Chaudhary, for, and even the students who have taken efforts to organize this webinar, and of course you all participants. I promise you that at the end of it you will definitely be more interested in knowing about cryoelectron microscopy. There are several papers in Nature, Cell, and you name any top journal, and you will see many of these papers on cryoelectron microscopy. So, what is cryoelectron microscopy? Like I mentioned, it's a mainstream technology for studying the architecture of cells, viruses, and protein assemblies at molecular resolution. Meaning, if you know about, or rather you actually know about Human Genome Project, and the kind of structural information, or rather the targets which can be employed or exploited for drug discovery, that's humongous. But then many of these targets, we do not have any information on the structural aspects, meaning we don't know how their three-dimensional structures look like. We know that we know their cellular localization, but we probably don't know their three-dimensional aspects. So that when it comes to structure function analysis, there are a lot of gray areas, and because of which utilizing these targets fully for therapeutic exploitation becomes a real problem. And that's exactly where structural biologists keep on coming with, like they keep they come up with many new approaches, wherein some or the other facets of the three-dimensional structure of the macromolecular assemblies can be revealed, and because of which we can use that information for basic discovery, like for basic sciences, as well as the applications including the drug discovery. And that's exactly where cryoelectron microscopy comes in. I mean, the word electron microscopy is famous, and we know at least the two versions of that, that is scanning electron microscopy and transmission electron microscopy, and that's exactly where cryoelectron microscopy fits in. It is a type of transmission electron microscopy, and as we move ahead you will know more about it, right? So like I mentioned, the recent advances, particularly in the computer or the image processing part, as well as the hardware part on the electron microscopy side, as well as the use of artificial intelligence and the deep learning, machine learning techniques have made it, they have taken strides to make, or to expand the applications of cryo-electron microscopy. And nowadays people are trying to use cryo-electron microscopy for looking at the interiors of cells, or the protein assemblies, which were which we could only think of in our dreams of looking at those. But now it is just becoming possible. If time permits, I'll show you the database as well, right? So this particular technique is routinely being used along with the conventional technologies. You may be aware of X-ray crystallography as well as nuclear magnetic resonance spectroscopy. These are the two widely used techniques for the three-dimensional structural determination of proteins or biomacromolecules, out of which X-ray crystallography has always been the front runner, while the nuclear magnetic resonance has been used to a great extent for looking at the solution structure, whereas the X-ray crystallography is for the solid state structure. But again, there are problems in the sense that X-ray crystallography cannot be really used for proteinaceous structures or proteins which cannot be crystallized, particularly those present in the membrane, because the moment you take them out of the membrane and the structure collapses, there is no way you could actually do something like do the structural determination. In fact, back in 2006, Brian Kobilka and his colleagues at UCSF, they actually got a Nobel Prize for coming up with a new technique for crystallizing membrane receptors, particularly G protein coupled receptors. So what they did was they chopped off the G protein part, or rather the intracellular loop 3, which is coupled with G protein of the GPCR, and then they replaced it with T4 lysozyme, which made it possible to prepare the crystals. And eventually they did the X-ray crystallography of beta 2 receptor, beta 2 adrenergic receptor, and for their work they got the Nobel Prize, I think 2 years later. So what I'm trying to tell you is the importance of like to be able to determine the 3D structure is central to structural biology as well as the basic sciences, and that's exactly why we are talking about cryo-electron microscopy. So having said cryo-electron microscopy, it's not just one technique, meaning it's a combination of various techniques which include I mean, of course, I will talk about I'll talk about these techniques, which include the cryo-electron tomography, single particle electron microscopy, and there are many others. Of course, I'm not going to talk about these because I did not want to make it complicated. But just I want to tell you that there are many divergence or many further extensions of the basic cryo-electron microscopy, which are now being used beyond what we thought we would use cryo-electron microscopy for. And that's exactly why it kind of concerns, or rather encompasses a large variety of techniques. But then the context is different. The basic principle remains the same, but the context is different, right? And that basic principle is imaging radiation sensitive specimens in a transmission electron microscope under cryogenic conditions. Now because this is electron microscopy, so we are using electrons as a source of radiation, and these high energy electrons which whose wavelength is of the order of say zero point zero 0.02 angstrom. If you are using a 300 kilovolt accelerating microscope, which is way too high, and these electrons are because it's a radiation, so they travel at the speed of light. So they when they hit the specimen, they actually cause damage. So the principle of cryo-electron microscopy revolves around doing the electron microscopy under cryogenic or at a much lower temperature, say liquid nitrogen or liquid helium temperature, so that the radiation damage can be kind of prevented, and the biological specimens can be observed at a very high resolution. And that's the basic premise of cryo-electron microscopy. right? So the applications of cryo-EM, apart from just looking at the protein molecules, people have used it for imaging intact tissues, particularly with the nanoparticles, as well as plunge frozen cells to individual bacteria, viruses, and bigger protein. I'm not talking about the smaller proteins like enzymes. You're talking about much bigger proteins. So if you look at TCA cycle, there are many protein assemblies or multi-domain proteins which catalyze variety of reactions in the pathway or in the cycle. So those multi-protein assemblies are the basic target of the cryo-electron microscopy, which otherwise is not possible with the help of X-ray crystallography, or even for that matter, the NMR spectroscopy. Okay, so this is what I was talking about. So cryo-electron microscopy, it encompasses the techniques such as cryo-electron tomography, single particle electron microscopy, electron crystallography, which are again subdisciplines of cryo-EM that have been successfully used so far for analyzing biological structures in different contexts. Meaning some of these have been used for looking at diseased tissues and the interiors of those diseased tissues or cells, and how do they compare with the normal cells, and there are many other different things. Now, having said that, if you look at the electron microscopy versus light microscopy, it's not very different in a way. It is microscopy wherein you can get to a larger magnification with light microscopy. It can you can go up to like 1000x or so for the obvious reasons, because the resolution, meaning how close like how you can see two objects which are closely spaced, it is the function of the wavelength used for the microscopy. So in case of light microscopy, we use visible light, and whose wavelength is 400 to 700 nanometer, or 4000 to 7000 angstrom. But in case of electron microscopy, like I mentioned, if you use an accelerating voltage of say around 300 kilovolts, you can go up to 0.02 angstrom or 0.002 nanometers. So you can imagine the resolution is not a limit. As far as, I mean, you can really go deep down at the biological level and take a look at it. But the whole problem is, because you're using high energy electrons which are traveling at the speed of light, they, when they hit the specimen, they actually cause damage, and this is where the whole limitation of electron microscopy lies. But of course, I have a video which talks about electron microscopy. So let me just try that video. If it plays nicely, I would definitely, it's just 2 or 3 minutes. It's a big video, but we'll watch for 2 or 3 minutes. So I'll just come out of this and share this particular part. Let me just go back. I'll put myself on mute, otherwise there will be echo. But just let me know if it is it visible. Is
Yes,
the
sir, it is
screen?
visible, sir.
Okay.
Yes, sir, it is
If
visible.
there is echo problem, just let me know. I'll just start playing it.
Microscopy, or SEM, is a powerful technique utilized in chemistry and material analysis that uses a scanned electron beam to analyze the surface structure and chemical composition of a sample. Modern light microscopes are limited by the interaction of visible light waves with an object, called diffraction. The smallest resolvable distance between two objects, or the lateral resolution, varies depending on the size of the diffraction pattern as compared to the object size. As a result, light microscopes have a maximum magnification of up to 1000x and a lateral resolution of up to 200 nanometers in ideal situations. SEM is not limited by diffraction, as it uses a beam of electrons rather than light. Therefore, an SEM can reach magnifications of up to 1 million x with sub-nanometer lateral resolution. In addition, SEM is not limited to imaging features only in the focal plane, as with light microscopy. Thus, objects outside of the focal plane are resolved, as opposed to light microscopy where they appear blurry. This provides up to 300 times increased depth of field with SEM. Chemists widely use SEM to analyze surface composition, structure, and shape of nanoscale entities such as catalyst particles. This video will outline the principles of the SEM instrument and demonstrate the basics of SEM sample preparation and operation in the laboratory. In SEM, samples must be conductive for conventional imaging. Non-conductive samples are coated with a thin layer of metal such as gold. Images are then generated by scanning a focused beam of high-energy electrons across the sample. The electron beam used in SEM is generated by an electron gun fitted with a tungsten filament cathode. The electrons are propelled toward the anode in the direction of the sample by an electric field. The electron beam is then focused at condenser lenses and enters the objective lens. The objective lens must be calibrated by the user to focus the electron beam on a fixed position on the sample. The focused beam is then raster scanned across the sample. When the primary electrons interact with the sample, they tunnel to a depth that is dependent on the electron beam energy. This interaction with the surface results in the emission of secondary and backscattered electrons, which are then measured by their respective detectors. The signal intensity of the emitted secondary electrons varies depending on the angle of the sample. Surfaces perpendicular to the beam release fewer secondary electrons and therefore appear darker. At the edge of surfaces, more electrons are released and the area appears brighter. This phenomenon produces images with a well-defined three-dimensional appearance, as shown in this SEM scan of asbestos. In contrast, backscattered electrons are reflected in the opposite direction of the electron beam. Detection intensity increases with increasing atomic number of the sample, enabling the acquisition of compositional information of a surface, as shown in this backscatter image of inclusions in glass. Now that the principles of the SEM instrument have been
Now let me come back to the presentation where I left. So the point of showing that particular video was to give you an idea as to what how electron microscopy works. So the overall principle of cryo-electron microscopy is also same, except the part that cryo part, what exactly they do, and that's exactly what we are going to see. So like we discussed, like we have seen in the video, the resolution is the key, and if you're talking about transmission electron microscope, wherein the wavelength for a 300 kilovolt accelerating voltage, it can be up to 0.02 angstrom, which is way too high, right? So because we are talking about transmission electron microscopy, the video was regarding the SEM, so don't bother that part. It is just the the electron microscopy part which I had to show, right? So when it comes to, like, after passing the electrons through the specimen, in case of transmission electron microscopy, these scattered electrons are focused by the electromagnetic lenses of the microscope, and eventually they are detected and then processed. The image is processed, right? So like I told you, resolution is a direct function of wavelength of the radiation, and it is not a limitation as far as electron microscopy is concerned, meaning the resolution. But then again, the problem is because of the high high energy electrons, the specimen is destroyed, or the radiation induced damage is a major problem, and because of which we cannot really image individual proteins, viruses, and cells in their native stage directly in an electron microscope at atomic resolution. And all the advances so far have always revolved around that. Of course, there have been advances in the electron microscopy part of it, or the hardware part of it, But then the major concern was the extensive damage caused to the specimen because of the interaction or due to the interaction of electrons with organic matter. So like we have seen in case of scanning electron microscopy, it is actually coated with a material, say gold splatter, or even for that matter substances like uranyl acetate, which eventually will will not we will prevent the damage to the organic matter. But then we are actually because we are coating it with some material, the organic matter or the specimen, so we cannot really get inside and look at, because that part is in a way covered. But that's where people try to come up with the say cryoelectron microscopy, or they try to look at solutions to this problem. Right, this I have already mentioned. The accessible molecular surfaces are coated with reagents which contain heavy atom, and in the last part of the video we have seen, as the number of like the atomic number increases, then the electron release from those particular, or the electron interaction becomes easy. But if you are talking about carbon, hydrogen, or the other commonly used atoms are present in the organic specimens, then in that case they are not really they don't really appear well. The resolution becomes a problem, and that's exactly where the major premise of cryoelectron microscopy lies, right? And that's where people have been using this particular mode of common mode of specimen, particularly for the conventional TEM, that is coating it with some heavy atoms, right? And people have been able to look at these microstructures to a resolution of 40 angstrom to 20 angstrom. So lower is this number, better is the resolution, and that's exactly what we would want to look at. So the now, one way of preventing the radiation damage is to use the lower electron dose. But then this lower electron dose results in images which have got poor signal to noise ratio, meaning the image appears blurry, and then those parts are not resolved properly. And that's where people try to balance the electron dose versus the radiation damage, and they just try to balance these things with. So basically, it's a trade-off between the electron dose and the subsequent damage, and that's where people try to come up with newer modalities or newer techniques, right? So people try to reduce the radiation damage which led to noisy images. But then the moment you go beyond certain electron dose, so then that is which gives you a good signal to noise ratio, but then the specimen is damaged, and eventually you don't really get what you would expect. And that's where cryoelectron microscopy came to existence, and that's where so we'll just quickly take a look at the basics of cryoelectron microscopy. So as the name cryo is low temperature, so here everything is same except the part that image is performed using frozen specimens maintained at either liquid nitrogen, which is minus 196 degrees, or liquid helium, which is even I think it is minus 269 degrees or so, right? Liquid helium temperature. And that those lower temperature basically is now that compensates for the radiation damage, so that because you are maintaining the specimen at a very very low temperature, say like minus 196 or so, so then in that case you can go with a little higher radiation damage without causing higher dose or higher electron dose, so that the specimen is prevented from the damage, right? So people have developed methods for rapidly freezing, that is vitrifying. The process is called as vitrification or vitrify biological specimens in a layer of glass-like ice. It is not it is not ice, meaning when you freeze water, this is an aqueous solution you are freezing, so you will get ice. But then they have come up with techniques which eventually prevents that ice crystal formation, but it's a little different state, and that is called glass like ice or vitrified material, which can be imaged at abnormally low temperatures, like liquid nitrogen temperature. Like so, they use something called as plunge freezing the aqueous solutions into a cryogen. Most of the times they use liquid ethane, which is further cooled by liquid nitrogen, and that's the interesting part. And one of those guys who got the Nobel Prize for cryo-EM, he came up with this single technique, and that guy worked for like last 20-25 years before he put forth this particular technique, which is now commonplace in cryoelectron microscopy. right? So here the imaging at liquid nitrogen temperature that reduces the extent of radiation damage, meaning you can increase the electron dose at the same time, so which will increase the signal to noise ratio, and because you are maintaining the specimen at say very very low temperature, the radiation damage is prevented. And that's what you can go up to like as much as six-fold. You can prevent the damage up to six-fold at the corresponding electron microscopy done at ambient temperature, right? So an image can be recorded at these abysmally low or cryogenic temperatures with much higher electron dose, and that's what has given the promise or the potential this technique has to offer, right? So another way to increase the signal to noise ratio that involves averaging images of a large number of identical units. And the third guy who got the Nobel Prize, or who shared the Nobel Prize for cryo, he actually came up with the way these images are processed. Meaning, I mean, in typical X-ray crystallography, billions of molecules which are present inside the crystal, they eventually are, their images, their diffraction patterns are averaged to come up with a single image, right? It's very interesting if you read more about how how X-ray crystal structure is elucidated, right? So what they did was they tried to use the similar principle wherein you average the images of a large number of identical units, and that's where the resolution went on improving. And believe me, it's for a drug discovery scientist like me, having an image, or rather the three-dimensional structure of a molecule at 1.2 angstrom resolution, this is like a dream. I mean, people have achieved this superatomic, or rather atomic resolution with X-ray crystallography, but with this cryo-EM technique, somebody has achieved this kind of resolution, which is amazing, and that's exactly why this technique is going to surpass. It has already surpassed the speed and the quality of X-ray structures, particularly for the assemblies which are not possible with the help of X-ray crystallography, right? And because of which, in a very short time, this technique has transformed into a mainstream method, and it has taken over the X-ray crystallography, particularly for samples which are difficult to crystallize, or those proteins which are membrane proteins. Like I told you, the moment you take them out of the membrane, the structure collapses, and you no longer will be able to observe their 3D structure. And there have been subsequent developments in electron microscopes, the detectors, as well as image processing tools, particularly the softwares, and that's where people have been able to achieve resolution up to 1.2 angstrom. Now I'll just take you to the regular workflow, and then I'll also show you the video. We'll finish in probably next 10 to 15 minutes, right? So I'll just briefly discuss, and then we will go. So what you have to do is you have to make a solution of a protein, or whatever you would want to visualize. You have to prepare the sample, right? And then the beam sensitive biological sample, which is preserved and immobilized in vitreous ice, the first part. So they use a grid or EM grid, wherein a very thin layer of the aqueous solution of the protein is put, and this grid is immersed inside liquid ethane, which is further cooled by liquid nitrogen. So you're talking of temperature somewhere between 160 to minus 160 to minus 196. So the moment that grid is immersed, eventually it becomes it gets vitrified, and that's what is subsequently subjected to electron microscopy, right? And this is the best part of it. So prior to preparing these cryo samples, one has to make sure that the sample is good enough for cryo data collection, otherwise all the efforts will be futile. And then there are various criteria for which by which you can check whether the sample is likely to give you a good cryo structure or not, or the data or not, right? Otherwise, it's a waste of time. So what they do is, the next thing, once the vitrification is done, they will subject that grid at very low temperature, like liquid nitrogen or liquid helium temperature, under electron microscope, right? So, and then the cryo-TEM imaging will be done, and the data will be collected. So this is like I would say a photographic film. So you have this grid placed, the electrons will come, and it's because it's a transmission electron microscopy, the radiation will pass through that, and then the image, subsequent image will be taken on that 2D projection. And then the most interesting part is the image processing, and in fact video will very nicely tell you. So the data that is recorded on a direct electron detector, usually in counting mode, and what this do is they usually take 30 to 50 frames, and there is again a particular requirement of a number of electrons per angstrom square area to be able to get the resolution which we require, right? So, and this is exactly what, like 50 electrons per angstrom square, to prevent the radiation damage, right? So again, these things have been optimized, and they, for a different specimen, like a cell versus a protein assembly, these electron doses will be slightly different, right? And then what they do is, the next part is, once the data is collected, the next part is the processing, right? So initially, just to see the feasibility, they'll work with a smaller data set to determine whether the sample has the potential for structure determination at high resolution or not. And so they will use techniques such as particle alignment and averaging, which is in a way, now because the electron beam is moving on the specimen, so this electron beam movement will cause movement in the macromolecule structure. So to negate the effect of the electron beam movement, they'll use these techniques to refine or to improve the quality of the image further. And then at the end of it, they'll get the 3D map, which eventually can be used for for taking a deep dive into the structure, just like what you would otherwise do for an X-ray crystal structure, that is a high resolution structure, right? So the images are analyzed by extensive digital image processing, and finally one or more atomic models of the structure will be built, which is actually shown here. There are these two videos. I'm just going to show you one after the other. These videos will make it very, very clear to you. The second video is, we will be, again, I mean, both the videos are nice. So what I will do is, I will take you to these videos. Let me see. I can directly go with that. Okay. I'm just going to play the first video.
Proteins are the building blocks of our cells. Understanding their shape and how they fit together could help us treat pain and even cure diseases. Scientists have been trying to get a good look at some proteins, but for years, the best we could see was a blurry blob. Many proteins are thousands of times smaller than the width of a human hair. Traditionally, scientists use a technique called X-ray crystallography to take pictures of the tiny proteins. For the X-ray imaging to work, it requires proteins are packed together into a stable, organized crystal. X-rays pass through the sample to create an image, but some of the body's most important proteins are too floppy or wiggly to line up into a crystal. Scientists at UCSF have developed a better way to view these elusive proteins.
If you have a floppy molecule at all, they just become more and more challenging to crystallize. Really, the problem was that these are big, complicated systems, and coming up with new ways of studying them required thinking about electron microscopy.
The reason cryo-EM works is that we keep the protein in a very thin layer of liquid, and we freeze them.
This technique is called cryoelectron microscopy. An electron gun shoots electrons at the speed of light passing through the sample. Then a specially designed high-tech camera captures the electrons to form an image.
The camera not only improve the imaging condition, give you much better sharp image, it can record a movie instead of steady picture.
If you had a still picture and I was doing this with my hand during the picture, I had a blurry hand picture. But with a movie, you could actually see each of the different steps.
A computer algorithm sorts the images and finds pictures of the protein in the same orientation. Then software builds a composite, an accurate high-resolution three-dimensional image of the protein.
And we're using the computation method to average many hundreds of thousands of molecules or tens of thousands together to generate a three-dimensional structure.
A structure like this one. Ifan calls it the wasabi receptor because it detects the pungent zing in wasabi and onions, and it's part of how the body registers pain. Scientists have known about this protein for years, but now we can finally see its shape in 3D. That means we can begin to build better drugs to block pain.
There is no audio to this video. Just keep on reading what is given. So I really hope you followed the process. So the first part was making a thin film on the grid, and then wipe out the excess liquid so that only a very thin film is left the left on the grid, and then it is flash frozen or plunge frozen inside by dipping it in the liquid ethane, and then that is subjected to the electron beam or picture by electron beam. So while it is flash frozen, the molecules are trapped in different orientations, and when the electron gun takes the picture, so hundreds of thousands of these molecules are trapped in the orientation in which they existed when the plunge freezing or vitrification is done. And then the next part is image processing, and then eventually you come up with these very very interesting pictures. Now, like I said, there is a database called Electron Microscopy Data Bank, just like the Protein Data Bank or RCSB, PDB. that is Research Collaboratory for Structural Bioinformatics Protein Data Bank. This one is Electron Microscopy Data Bank. So if you look at the structures, so in recent times there have been now you see on 6th October 2021, the EMDB contains 16,980 entries, while the entries in PDB are close to 200,000. So we are talking about the X-ray crystallography structures which started back in like 1978. In last 44 years, only 200,000, and this is probably in last, I would say, less than 10 years already 16,000 entry. So the speed is tremendous, and eventually the structures which have been determined, like you can go and search in there. So those structures are really complicated, and there is no way we can actually do similar structure determination using 3D like X-ray crystallography, although we talk about high throughput X-ray crystallography and all, but then those membrane proteins or those multi-domain proteins, it's impossible to really determine their structure using X-ray crystallography. And that's where the electron microscopy like cryo-electron microscopy comes to so these are the three guys who actually contributed to the stage of the art of I would say cryo-electron microscopy. Henderson, he has been working on the 3D structural determination of proteins since what 1970s or maybe little before that. Frank was the one who came up with that image processing stuff, and then Jack Dubochet, he came up with that technique, that vitreous ice formation technique. So they contributed to this discovery, and because of, I mean, they worked independently. Believe me, it wasn't that they worked together or something. They worked independently, but then at one point of time they just ended up using the discovery done by other, and that's exactly how this cryo-electron microscopy came to existence, and that's why they were awarded the Nobel Prize in Chemistry in 2017, which is the highest honor any scientist can ever get. So these are some of the pictures from their Nobel lecture. You can go to the website, Nobel Prize website, and you can search for this, and you will get a full transcript of what they said. Amazing stuff. You should read about that, right? So these are very, you can see how complicated these proteins are. So these are the individual assemblies, multimeric assemblies, and this is the top view, and this is the side view, like that, and this is the actual protein. And this is how, like, resolution before 2013, where we just saw the blobs, but nowadays the resolution is very, very high, and you can see individual side chains or atoms inside the protein, which is what we are most interested in. And this is what revolutionized or going to revolutionize or already started to revolutionize the structural biology arena and the overall drug discovery arena. Right, so this was about cryo-electron microscopy. So again, the same question: isn't it amazing? And I'm sure your answer is yes. So with this, I thank you for your patient listening. And if there are any questions, I would love to take those questions.
Uh, Thank you, sir, for this wonderful presentation, and especially the selection of the video. I must say, I have been teaching them microscopy, but I never came across the first video that you showed, even the subsequent one. They were excellent. I'd be very happy if you could share them, if it is available
Yes,
in,
yes, ma'am.
open source. So if you could share them, because that's a wonderful material. Thank you for this excellent presentation. I'm sure my participants have also enjoyed that. And thanks for accepting our
Thank you
invitation.
so much, ma'am.
Now, as I mentioned, that we have included this as a part of our ongoing curriculum also. So, students, the question for today is, just sum up what you have been able to understand in terms of the presentation that you had today from Professor Kharkar, and within 30 minutes, send your responses to my email. So, thank you, stay safe, and we'll again meet with our fourth talk, perhaps, as I remember, it would be on 30th of October. So, till then, stay safe. Thank you, everyone.
Thank you so much. Thank you so much, ma'am. Take care. Bye.